All right, guys, happy Thursday. I'm home with my kids today. But we are going to talk today about the actual phases of mitosis. Quick review that yesterday we talked about how before any complicated cell like a eukaryotic cell divides, it's got to do two things. It's got to organize and copy its DNA. Well, copy and organize its DNA. And that not all cells are dividing in your body at once. You might find lots of division in skin cells or maybe your bone marrow. But a lot of other places, cells are kind of just hanging out in some place in this cell cycle that we talked about yesterday. That cell cycle consists of course, mitosis, but beyond that, when the cells come out of mitosis, they got to grow, they got to get back to normal size. That's G1 phase. They got to copy their DNA to prepare for another mitotic division. That's S phase. And then G2 is making more cell parts and growing a little bit more for preparation for the next mitosis. Then mitosis has the phases prophase, metaphase, anaphase, and telophase, with the end of it being cytokinesis. So we're going to look at each of those phases today, kind of what they look like, and then I'm going to have you go hunting on the internet for photos of those actual phases in preparation for you seeing them for real. Just a last bit of review is that those cells that you have are regulating their mitosis, regulating their cell cycle with these checkpoints, one at the beginning of G1, one at the end of G1, one at the end of G2. If they don't have the stuff they need to go into a new division, they stop in one of those checkpoints and just kind of do what cells do. So moving on to the phases of mitosis, which remember are prophase, metaphase, anaphase, and telophase. But of course, there's also that in-between phase when a cell is in G1, S, or G2. We call that interphase. So you should draw each of these in your notes. And in interphase, we see what I call a ball of yarn, right? That's the chromatin. It's unfurled and doing all the stuff that DNA is supposed to do. We have a distinct nuclear envelope, and then we have the outside cell membrane. And one of the other things that we kind of skipped over when we talked about cell parts are centrioles. These little cylinder-shaped things here are going to become real important as mitosis gets started. So the first and last phases are the most complicated. Starting with prophase, what we find in prophase is that this dashed line here means the nuclear envelope is going away. It disappears. At the same time, the chromatin is condensing into chromosomes. Okay, that yarn-like stuff is condensing into these X-shaped bundles, and the centrioles are starting to grow this thing called the spindle. The spindle is starting to grow between the centrioles. That's going to spread out to either end of the cell and wind up grabbing onto those chromosomes. The nuclear envelope's disappearing, like I said, and the chromosomes are starting to make their way towards the center of the cell. That's prophase, basically real busy phase where everything's getting ready for that division of the nucleus. Remember, we said that the nucleus is what's really dividing in mitosis, not the whole cell. We're really concerned with what's going on in the nucleus. So the first thing is the nucleus basically disappears. The chromosomes get made. The spindle gets built between the centrioles. You'll see what happens with that, and the chromosomes start moving. That phase lasts a while. Then there's kind of a split second phase where all those chromosomes, from being jumbled around in the cell, they line up down the middle. Right? They line up down the middle, and the spindle gets finished and attaches to, remember, that's called the centromere of each chromosome. So in metaphase, the chromosomes line up in the middle. The spindle fibers finish attaching to the centromeres of the chromosomes. Sticker question that I'll come back to on Friday. What do you think spindle fibers are made of? First one to get it on Friday will get a sticker. So metaphase doesn't last long. It's just when those things are lined up down the middle. So go ahead and draw that. Moving on, as soon as you see daylight between the sister chromatids, because you knew that was where this was going, right? Those X-shaped things are splitting in two. As soon as you see space between them, it's not metaphase anymore, it's anaphase. Anaphase, the sister chromatids start to separate, they move away from each other, and the cell membrane actually does sort of start to pinch in here. And I'll show you this better on Friday when I have the board to draw on. But here and there, you start to see just a little pinching in there and there, as the cell starts to do this thing called cytokinesis, where it's eventually going to actually divide the cytoplasm. That's really a separate event from mitosis. Remember, mitosis is the division of the nucleus. So that's anaphase. As soon as they separate, and until you really start to see deep creases in the top and bottom of the cell membrane, that's anaphase. And telophase, you can see what I was talking about these creases. Telophase is the other really busy one besides prophase. What we see in telophase is the sister chromatids moving even further apart. We see the new nuclear envelopes forming here and here. We see the nucleoli reappear, those dense spots in the nucleus. I didn't draw those, but you know they're there. The chromosomes actually toward the end start to uncoil and get all blobby and yarny again. And the spindle fibers and the little stuff coming backwards called asters, those start to get broken down as well. So that's telophase. The cell membrane continues to pinch in, okay, through telophase, began in anaphase, it keeps pinching in telophase. At this point, when we have two separate full nuclei, mitosis is basically over. But there is one more event, and that event, when we have two new cells, is called cytokinesis. That is the division of the actual cytoplasm into full cells. Remember, it started during anaphase. It pinches in until the cytoplasm separated. In animal cells, in plant cells, it's a little different because there is, of course, a cell wall, right? And that cell wall isn't going to pinch, it's kind of stiff. So what happens instead is you kind of just build sort of a hairline crack in between to define those two cells. And the new cell membranes and cell walls grow in that hairline crack in between the two cells. So that is a quick run through the phases of mitosis. If you didn't have time, you know, use that pause button, get those things drawn in very well in your notes. And so, yeah, your assignment today, as you look at those phases you've drawn in your notes, is going to go on an internet scavenger hunt. And in this scavenger hunt, you're going to be creating a Google Slides presentation. It's going to have 5 slides, one for interphase and one for each of the 4 phases of mitosis. Each slide needs to have the name of the phase, 2 photos from the internet that you copy and paste in, one from a plant cell doing mitosis, one from an animal cell doing mitosis, and a list of all the events from your notes that are happening in that phase. This is an individual assignment, so please don't work with anybody else. Please don't use any photos that are already paired up for you on the internet. You know, like there's, you know, plant cell and an animal cell next to each other. I want you finding them individually. And the URL, the website you used for the actual photo, should be pasted under each photo. That is your assignment. That assignment is actually due on Monday, but you have the whole period today to work on it. You probably won't get any time tomorrow to work on it, so make sure you get some good work done today. I hope you're having a great day, and I will see you in person tomorrow. Bye, guys.